Hi, I'm Steph Strickland with GeekWire Studios, your host for Does Compute, the podcast where we delve into the latest innovations in computer science with the help from experts at Carnegie Mellon University School of Computer Science. I want to introduce you right now to Dr. Oana Carja, an assistant professor in the Ray and Stephanie Lane Computational Biology Department at CMU. Her research focuses on understanding the evolutionary architecture of intelligent collective systems by utilizing tools like dynamical systems, network theory, population genetics, machine learning, and statistical inference. She often leverages, and we'll talk about this more specifically, widely available yet underused data sets in her work. Dr. Carja, essentially, you sit at the intersection of computational biology, evolution, complex systems, and mathematics. I make it sound so simple. It's not. Do me a favor. Tell people about what got you interested in computational biology. You didn't start with a love for biology necessarily.
No, that's true. I grew up really loving mathematics. I was fascinated by the rigor and the process of mathematics, the beauty, the elegance. I started going to math Olympiads, and even my undergrad is in pure mathematics. And then I got into biology quite by chance. It was actually the first time I came to the US. I applied for a summer internship to do undergraduate research, and I ended up at this institute in Santa Fe called the Santa Fe Institute, which is an institute for complexity science. And there I was just very randomly paired with some scientists from Los Alamos, a biologist and a biophysicist. And they were working on a very simple, impactful problem. Let's come up with a vaccine for HIV. So that summer, starting to work with them was really my introduction to biology, to genetics, to understanding about the DNA code, about evolution as a process connecting every living thing. Before that summer, I had often thought, oh, biology is just memorization. It's messy. There's no beauty to it, like in mathematics. But that summer, and then my subsequent PhD and career in the field taught me how much that is actually not true, that biology is far from just being memorization, that there is process and elegance and rules in biology too. And that was just absolutely fascinating to me.
This has to have really changed the game for you to have AI and machine learning and the ability to dissect patterns in these large data sets that have been available but perhaps underutilized. This had to have really excited you as someone who describes mathematical processes as elegant and beautiful. Talk to me a little bit about that aha moment when you really understood what math could bring to this broader discussion of biology and evolution.
Absolutely. It's interesting that you mention AI, and I completely agree with you. But at a first glance, one might think, well, AI is black box. It doesn't really help us understand mechanism and rules, right? We'll see that is actually that's actually not true. So what I particularly focus on is essentially trying to understand a little bit more about the process of evolution, right? As I mentioned, everything that has ever lived and is living and will ever live is shaped by the evolutionary process. And that just like blows my mind, right? And again, while most people tend to think about the evolutionary process as this very long term process, right, over millions of years, or the out of Africa expansion of modern human colonizing all these different parts of the world, evolution can also happen very fast. And in fact, some of the most pressing failures in our medical system are actually driven by a process of fast evolution, right? So, for example, drug-resistant bacteria, tumors inside of our bodies, treatment failures, all arise from the process of evolution and from the fact that these bacteria or these cells, these groups of cells are constantly evolving and evading our weapons and our treatments, right? Whatever we throw at them. And so I became very fascinated by that, and by the fact that, we think we know the rules of evolution, right? Since the time of Darwin. You have heritable variation upon which natural selection acts. But in fact, there's still so little formalism when it comes to us understanding the rules of evolution. There is still so little we understand about the commonalities and the differences between different systems. And why do we want to understand that? Well, the hope is that by understanding how these systems evolve, take systems like viruses, right? How does COVID evolve? How does a virus like COVID evolve? How do populations of bacteria escape treatment? And so you have this public threat of antibiotic resistance. How do tumor cell populations evolve that allows some patients to have successful treatment and some patients to simply not respond to treatment, right? All of these, if we understand these rules and these systems better, the hope is that we can then, in turn, build better weapons in order to combat some of these failures in treatment. And so what I find particularly fascinating, going back to your question, is that both in terms of mathematical tools as well as computational tools and now machine learning, biology is transforming itself into a quantitative science. And so we're bringing these tools to the process of evolution to essentially better understand what drives the evolution of these systems. And I think it's absolutely a fascinating time in computational biology right now, because again, we often tend to think of biology as messy and the exception to the exception to the exception, and not as quantitative as physics. And what I think is fascinating right now is that you do have this field undergoing an utter transformation in terms of what you can discover, driven by these new technologies, right? Driven by our ability to process data at scale, to collect more data, to have these algorithms help us make sense of these big data sets.
It's interesting to me when you talk about evolution, because you're describing at its core some of the smallest building blocks of life as we know it. Yet when I think about the way you describe it, it reminds me of our understanding of space and the universe beyond. As we learn about new aspects, it completely changes the mathematical foundations of what we thought we understood. And it applies to some of the direct disease research that you're doing, breaking down things at the atomic level, understanding how cells divide and mutate. So with that said, I want to talk to you about liquid biopsies, because I think it is probably the most articulate way to begin this discussion about how our understanding can really shape in the development of new weapons to combat cancer. Let's talk about liquid biopsies.
So what is liquid biopsies, right? Imagine a world where you could simply take blood from a person, so a very uninvasive method, and looking at the DNA that is present in this blood sample, you could be able to say, well, does this person have cancer inside their bodies? Or does this DNA help me better understand the tumor? Let's say we already know that this patient has a tumor in their bodies. Does it help me better understand what is going on inside the body of a patient? Does it help me understand if the cancer has returned, right? What treatment should I use for the patient? So while this technology, this liquid biopsy, or another way to people refer to it as understanding circulating tumor DNA, right, in the sample of the blood, while this technology, it's not hard to explain why it can be transformational. It's still very far from its promise. There are still many different reasons why It's very hard to understand how to make use of this data, these DNA sequences in the blood, to understand what's going on in the patient. And another way to look at it, imagine almost like you have the tumor inside the patient and a mirror, right? That and then you have what's in the blood of the patient. So imagine this mirror. You're essentially trying to understand, how does the blood mirror the tumor inside the patient? What we're working on and what we're trying to understand is how this mirror might be distorted. The reason why liquid biopsies or circulating tumor DNAs are far from reaching their potential is because this mirror is a very distorted mirror by some of the processes that are happening inside the patient, some of the evolutionary processes. Some clones more predominantly end up in the blood than others. Some clones migrate more than other. Natural selection makes some clones be more abundant, and that may or may not be represented in the circulating tumor DNA. So what me and my groups are working on is trying to understand, how might this mirror be distorted? How does the evolutionary process, the tumors as these evolving ecosystem, how do the processes in the tumor microenvironment inside the patient contribute to the distortion in this mirror? Because if we understand the distortion, then we can correct for the distortion such that this mirror is more faithful. So we're working together with physician collaborators, surgeons, trying to essentially having data set of both what's happening in the blood of the patient, different samples of the main tumor, images of the tumor, to essentially try to bring these disparate multimodal types of data sets together to help make headway into this very complex problem, right? And again, cancer is as complex as it gets, is this incredibly fast evolving, complex ecosystem. And that is why some of these novel tools, together with, what I want to emphasize, it's some of these complex ecosystems, making headway into these problems can only happen when you have very interdisciplinary teams together with novel, powerful tools like AI and machine learning to help us really solve these difficult problems.
I wrote that down because I want to come back to it. When you describe this interplay between what's happening in the tumor and the ability to focus the mirror, you're describing glasses. You are trying to fix the vision so that it is a straight across, and it's needing to understand where the distortion happens so you can correct for it. It's a gross oversimplification, but the work is fascinating nonetheless.
Absolutely. It's essentially, hey, I want to see inside the body, right? Help me. How do I build good glasses to see inside the body? Absolutely.
So we've talked about liquid biopsies and the ability to peer inside the human body, and also just peer inside the mathematical structures to glean out the consistencies in the data. How do you even begin to tackle something like this from a mathematical perspective? We've talked about the fact that you will utilize these data sets that are underutilized. Are you going back to what's already out there? How does this process work?
This is something that comes with a lot of experience too. And again, I think interdisciplinarity here, attacking the problem using many different types of skills and approaches, is very important. When it comes to, so in my group, we build a lot of mathematical models, as well as algorithms to tackle different big quantities of data and different types of data sets. When it comes, I actually teach 2 different classes at CMU on mathematical models. How do you build mathematical models for biology? How do you build models in general, be it mathematical or computational models for biology? My first class is just general biology, and my other class that I teach in the spring is called Modeling Evolution. How do we uncover the rules of evolution using mathematical and computational models. And something that the art of modeling that we try to teach our students too really lies in how do you understand and capture the relevant properties of a system and incorporate in the model only what's necessary and what is useful, and how do you learn what is superfluous so that you get rid of it, right? The art of mathematical modeling and building elegant, simple enough mathematical models really lies in trying to find the drivers of the system, and learning what you can ignore, right? The properties that just simply don't matter that much. And there's this particular sort of tension when you build models between, hey, I want to really understand mechanism and build simple enough models, versus trying to incorporate too much. And building good models really also depends on your goals. Sometimes you want to optimize for generality. Sometimes you want to optimize for realism and precision. So there's all these different goals and subtleties when it comes to building mathematical and computational models. I think the same is true when it comes to collecting the data sets and deciding which data sets to use and to link to your mathematical and computational models. Why I often say that the data sets that we use are underutilized is because we often, in a lot of our work, it's true that in some cases we also build our own data sets. We actually do experiments in the lab. We collect data from patients in a very directed way. But we also, in a lot of our work, use public data sets, right? Data sets that were built for other questions. And we find ways to squeeze more information from those public data sets that people simply haven't squeezed before. A lot of these data sets are, for example, a lot of spatial data sets, because imagine, I mean, it's the same in other fields, right, where AI is having a lot of impact. Now we talk a lot about text, but initially it was images, right? AI being able to process huge amounts of image data at scale, and the availability of new technologies that allow us to have spatial image information in terms of what's going on at the molecular or cellular level, for example, in biology, right? How are molecules inside the cells coexisting? How are cells inside tissues coexisting, communicating with each other? All of this has just been available only in the last few years. And it's absolutely spatial data sets, the ability to process them at scale has absolutely transformed cellular and molecular biology. But these data sets have not really been utilized for making progress in evolutionary biology. So this is sort of where we essentially bring a different perspective to analyzing some of these data sets, or utilize them not for their original purpose, but there is a certain amount of creativity that goes into picking the right data sets to answer questions in a very different field.
I want to touch on something you mentioned before I come back to evolutionary biology and the speed with which we can look at evolution in antibiotic resistance. But you've mentioned twice now this sort of collaborative approach for your work at Carnegie Mellon. And I want to talk more about that because every professor and researcher I have spoken with has mentioned that as being something that is unique and special about the learning and the research that happens at the university. Talk to me a little bit about the interdisciplinary approach that you're able to take as a result of where you were teaching.
Okay, so basically, I'm not saying anything new, right? Like it's true. I think some of the biggest progress in science has usually been made when people from different disciplines come together and learn from each other and bring their own expertise to tackle hard problems that neither of them could independently, right? And I think this is also kind of a little bit what's very exciting to me about the as a sort of side note about the promise of AI as a potential collaborator in this process, right? We're not there yet, but wouldn't it be cool if we had a little AI agent helping us, essentially a scientist, learn differently, ask better question, ask targeted question when it comes to these systems where it's simply really hard to make progress alone. Right. I think my experience when it comes to collaborations, again, was informed in that very first research experience I had in when I was an undergrad at with the two researchers from Los Alamos in Santa Fe. I was paired with a biologist and a biophysicist. And so it was absolutely fascinating to me how these two very different researchers had actually just recently started to work together, brought very different domain expertise. The biologist, Betty Korber, was this renowned HIV researcher. And the biophysicist, Tanmay Bhattacharya, was basically the head of biophysics at Los Alamos, bringing in all this quantitative expertise, computational, mathematical expertise. And they essentially were trying to tackle this very complex problem of trying to come up with a vaccine for HIV by bringing that expertise together. And what was fascinating to me about it was how they almost were essentially trying to find a common language, right? I was there in meetings with them. And this is, mirrored at my meetings now with collaborators at CMU, where the biologist would say something about what might be important for that system. And then the quantitative people in the room would almost try to translate it in terms of their language and try to see, well, what might that mean and what tools can we bring to the table? So it was a constant sort of attempt at finding common ground and communication. And only through that joint sort of understanding and communication can you have this outsized impact in a project. And so that experience, again, was absolutely instrumental for me as an undergrad. And I've seen it over and over again in my career, because while we say collaboration is essential, especially for these very hard problems, very complex problems, it's also true that collaborating, finding common ground, finding that common language, right, is incredibly hard, because for better or worse, we are still taught across in these very different disciplines, and it's actually very hard to find that common language and common ground, right? We're still kind of siloed in our respective departments. We're used to particular nomenclature. We have our particular language, right? And that's not to say that it's a bad thing. I actually think, something that I just taught in class the other day was this lecture on cultural evolution, and actually the evolution of language. We have so many different languages in the world, and languages are constantly evolving. And if you think of mathematics, right, having its own language is actually one of the most efficient languages in the world. So the fact that every field has its own language is not a bad thing. But it's fascinating to me how you almost, have to create and learn a new language as new fields and new backgrounds come together. And this allows us to make a lot of progress on these very hard problems.
Outside tumor modeling and understanding that is still challenging, even though the work in the field's not new. It might be growing exponentially. We've been thinking about disease for quite some time in that fashion. But I want to talk about antibiotic resistance? Because even the layperson or someone just getting a prescription for an antibiotic has some understanding of that. How does your research shape the discussion around solving for that problem?
There's many different aspects that we are tackling when it comes to something like antibiotic resistance. First of all, we're even trying to understand why we might see different rates of resistance in hospitals or in vivo compared to the expectations that you see in the lab. So something that I don't know if your audience knows is that rates of resistance when you're experimenting with these bacterial populations can be incredibly different in the lab compared to what we see in the hospitals in the real world. There's many different sort of angles to try to understand why, right? Because again, when you don't understand a phenomenon, you're kind of trying to feel your way towards seeing, well, what kind of model might fit this data? And one particular approach that we have and that has shown success is trying to understand how spatial structure potentially can affect not only the rates of resistance that you see in real populations, but also why might you see something that has puzzled researchers for a long time now? Why might you see sensitive bacteria coexisting with resistant bacteria, almost like in this happy collaboration and coexistence, right? Because think about it, right? It's very easy to imagine, not knowing much about evolution, but we know about natural selection, right? Survival of the fittest. One might imagine, well, resistant bacteria is just better, right? It's able to resist antibiotics, right? It's able to grow even in the presence of treatment. So if you technically have something that is better, right, that divides faster, just grows faster, why would it not simply drive to extinction the other population, the sensitive bacteria, right? And yet what you see is sensitive bacteria coexisting with resistant bacteria. You see these unexplained different rates of resistance in different populations. So part of my group is building mathematical and computational models to try to understand how spatial arrangement and the topology of the systems in which bacteria grow affect these rates of coexistence and affect rates of resistance. Even though the systems are very, very different, right? Bacteria and cancer, understanding there are certain things about the rules of evolution in bacteria where the hope is that maybe there are commonalities, right? Maybe if we understand how sensitive and resistant bacteria can coexist with each other, maybe potentially might help us understand what's going on inside a tumor where you also have resistant mutations potentially collaborating and competing with a normal, the normal tumor population, right? The sensitive tumor population. It doesn't always apply, right? It doesn't always port. Some systems can be very different from others. But what's beautiful about evolution is that it does provide unifying mechanisms, right? The forces are the same. You have the force of natural selection, mutation, recombination, drift, and migration, all fighting together to shape the compositions of these populations.
This discussion, and I love how you brought it full circle, be it tumors, antibiotic resistance. I can see this extrapolated into pathogens and beyond. So if at its core, it's really understanding spatial structures, what do you hope your broader vision as we wrap up our time here? What is your biggest open question then that you want to tackle, given how many aspects of life and existence can be touched by an understanding of these spatial structures?
At a very big question, right? For now, we are very focused on essentially understanding what are the shapes of these systems, right? If we are trying to think of spatial structure of this, the shape of the system, the topology of the system, of individuals in the population essentially co-localizing, communicating, almost like, you have the social lives of bacteria, the social lives of tumor cells, meeting, exchanging genes, migrating, right, moving in the population. At a big picture level, what we're trying to do in my group is try to understand what are these different topologies in real world systems, and how has evolution shaped these topologies, but more importantly, does the topology of a system shape its rates of evolution, right? Can we understand how topological properties affect rates of evolution? When are you more likely to see resistance? How is it likely to affect what you see in the blood? When are you more likely to see coexistence, right? So at a very big picture level, we're both trying to understand what are the topologies that you see in the world. Another example is, what is the effective social network on which a virus like COVID is spreading? How is that changing over time, right? If we think about viruses being transmitted between different hosts, right, between different humans in a population, you can imagine that if we were all well mixed, right, a population that is all very crowded, right, and very connected to each other, potentially allows for a different evolution of the virus, right? As the virus keeps moving between individuals, mutating in the population, that allows for a different rate of evolution for a virus than if the population is very fragmented, right? If it's very hard for the virus to spread through that population. So I think that is usually a metaphor that I use in my work, because it's very easy to imagine how the spread of viruses in today's connected world, when we have airplanes and all kinds of different amounts of connectivity in our population, is very different than, let's say, 100 years ago, when the connectivity of the population was very different. Essentially, we don't even know what is the effective topology of spread for a virus like COVID, right? So if we were to understand that, and if we were to have a unifying theory of how population structure affects rates of evolution, then we could potentially try to understand the commonalities and the differences between all these different systems and what affects rates of fast evolution, right? So another way to think about it is, I've told you that rapid evolution drives disease, right? And there are hints that spatial structure drives rapid evolution and is a major contributor into rates of rapid evolution and how rapid evolution proceeds. And so the hope is that by understanding spatial structure, we're able to better understand rapid evolution across all these different systems.
It's the social network at the atomic level, really. This has been so enlightening. Dr. Carja, thank you for taking the time to talk to me today.
Thank you so much. This is fun for me too.
Everyone, you have been listening to Does Compute, the podcast where we examine how computer science is building useful stuff that works. Thanks so much again for joining us. I'm Steph Strickland with GeekWire Studios.